Everyone, welcome to my channel Anupma Biology Classes. This is the third series of my classes, that is the reproduction. Today's video we will discuss about the gametogenesis. It means gamete formation, and this video is from chapter 3 part 5th of CBSE class 12th. So here the first topic is gametogenesis. So what is the meaning of gametogenesis? Gametogenesis is the process whereby a haploid cell is formed from a diploid cell through meiosis and cell differentiation. In case of male, it is known as spermatogenesis and produces spermatozoa, while in case of female, it is known as oogenesis and results in the formation of ova. Now, the phases of gametogenesis. In both cases, that is in spermatogenesis and oogenesis, there are three phases. Here, the first one, that is the multiplication phase, in which the germ cells of the gonads multiply by mitosis. Second is growth phase, in which the germ cells grow in size. And the third is maturation phase, in which the meiosis takes place to produce the gametes. Now, here we will discuss and understand every process in detail. So, in spermatogenesis, sperm will be formed in testis, and in oogenesis, ovum will be formed in ovary. Now, the first gametogenesis, that is the spermatogenesis. Spermatogenesis takes place in the seminiferous tubule of the testis. Seminiferous tubules are lined by germinal epithelium. It consists largely of primary germ cells and certain Sertoli or nerve cells. Now, the process of spermatogenesis. If we talk about the process of spermatogenesis, we know that it is a continuous process, but for convenience, it is often divided into two stages, that is, formation of spermatids and formation of spermatozoa. Now, the first stage, that is the formation of spermatids, which you can see in the picture. It has three phases. First one is multiplication phase, second is growth phase, and the last, third, is maturation phase. So in this picture you can see here the first, that is the multiplication phase. Here at maturity, the primordial germ cells divides by mitosis to produce a large number of spermatogonia. And this spermatogonia have prominent nuclei that contains diploid number of chromosomes. Like in humans, 23 pair of chromosomes present, it means 46 chromosomes in number. Now, spermatogonia increase their number by repeated mitotic divisions with same number of chromosomes and also provides growth of the testes during early life of the animal. Now, the second, that is the growth phase. Here, on sexual maturity, some spermatogonia stop dividing and grow in size by accumulating cytoplasm and replicating DNA. And this spermatogonia with a large size, we say it primary spermatocyte, and this growth phase is called spermatocytogenesis. Other spermatogonia which are not involved in growth continue to divide mitotically and produce more spermatogonia for later spermatogenesis. After the growth phase, the next is the maturation phase. And in this phase, the primary spermatocyte by the meiotic division form secondary spermatocyte. And this is the reduction division. So the diploid primary spermatocyte is converted into haploid secondary spermatocyte. Now this secondary spermatocyte undergo meiotic divisions to produce haploid spermatids. Because of this, sperm has only half number of chromosome like in male, which have only 23. After this, the last stage is spermatozoa formation or spermiogenesis, and here after the metamorphosis, spermatid converted into sperm or spermatozoa which are haploid. Now, here we will understand the complete process of the spermatozoa formation. In this picture, you see the spermatogonia form primary and secondary spermatocyte, which forms spermatid, and then spermatid. After metamorphosis, the spermatid forms spermatozoa. So here, from one spermatogonium, four haploid spermatozoa are formed. Spermiogenesis is the final stage of spermatogenesis, which sees the maturation of spermatids into mature spermatozoa. It involves DNA condensation, replacement of nucleosomal histones by protamines, and loss of virtually all cytoplasm with development of the proteolytic enzymes, rich acrosome cap, and a flagellum capable of propelling beating movement connected by a mitochondria-rich midbase. So, the development of sperm cell takes about 2 months and the fully formed sperms become free in the cavity of seminiferous tubule. The process of release of spermatozoa from the Sertoli cells into the cavity of seminiferous tubules is called spermiation. And from here, the sperms passes via vasa efferentia into the epididymis for temporary storage, and here they acquire motility by testosterone. Now, in this picture, you can see the whole process where it occurs. At first, you have to clear that the sperm formation occurs in the testes in seminiferous tubule, and this process is called spermatogenesis. Here, the cells of germinal epithelium shows many steps like multiplication phase, growth phase, maturation phase, form spermatids which after metamorphosis form mature sperm. And here two meiotic divisions and one mitotic divisions occur. So, it is a highly organized process that produces millions of sperms each day in the postpubertal mammals. And this productivity is dependent on the activity of spermatogonial stem cells. And here, the adult tissue stem cells in the testes that balance self-renewing divisions with differentiating divisions, which maintain the stem cell pool and fuel spermatogenesis respectively. When spermatogonial stem cells differentiate, they give rise to spermatogonia, which undergo a species specific number of transit amplifying mitotic divisions, which followed by two meiotic divisions and spermatogenesis to produce terminally differentiated sperms. So, this is the complete process of spermatogenesis, which has two main processes, that is spermatocytogenesis and spermiogenesis, that means formation of sperms. Now, what is the difference in the spermatocytogenesis and spermiogenesis? So, spermatocytogenesis is the second phase of the spermatid formation, while the spermiogenesis follows spermatid formation. Spermatocytogenesis produces diploid primary spermatocyte from spermatogonia, while the spermiogenesis produces mature haploid spermatozoa from spermatids. Spermatocytogenesis involves merely growth of spermatogonia, while the spermiogenesis involves movement of cell organelles to positions characteristic of spermatozoa. Spermatocytogenesis, here the cytoplasm is added and the DNA replicates, while in the spermiogenesis, a good deal of Golgi apparatus, cytoplasm and water is lost. Spermatocytogenesis is followed by many events, while the spermiogenesis is the final phase of spermatogenesis. So this is the main differences in the spermatocytogenesis and spermiogenesis. Now, what is the role of Sertoli cells? So, during spermatogenesis, the germ cells are closely associated with the Sertoli cells. Spermatogonia lie between the Sertoli cells and the basal lamina. Other stages occur in the pockets along the sides of the Sertoli cells. Sertoli cells provide mechanical support, protection, and nourishment to the developing spermatozoa. All nutrients, gases, and the waste exchange between the developing spermatozoa and the blood vessels around the seminiferous tubules pass via the Sertoli cells. In this picture, you can see the Sertoli cells and their position. So, at all the stages of differentiation, the spermatogenic cells are in close contact with the Sertoli cells, which are thought to provide structural and metabolic support to the developing sperm cells. These cells are characterized by vesicular, oval, basally positioned nucleus, which contains a prominent nucleolus. Sertoli cells are endocrine cells. They secrete the polypeptide hormone, that is inhibin, which acts as the label of the pituitary to reduce the secretion of FSH, that is follicle stimulating hormone, and it shows negative feedback of the hormonal action in male, which we discussed in the previous video. Now, the structure of spermatozoa. Here it is a structure of a complete spermatozoa which has four parts. First is head, second is neck, third is middle piece, and the last is tail. So, in this picture, this is the complete structure of a sperm. And here the first, that is head, which is composed of nucleus and the acrosome. Nucleus has only condensed DNA and the basic proteins, while the acrosome formed by the Golgi complex and contains hydrolytic enzymes which is used to contact and penetrate the egg in fertilization. Second is neck, which is short and contain two centrioles lying one behind and at right angle to the other. Here, the proximal centriole plays a role in first cleavage of zygote, and the distal centriole give rise to axial filament of the sperm. So, after the formation of axial filament, it degenerates at mature stage, and that is the proximal is present in the mature stage, not distal centriole. Now, the third part, that is the middle piece, which contains mitochondria, which provides energy for the movement of sperm in the female genital tract. If a sperm fails to contact an ovum within a specific period, that is 24 to 48 hours, it exhausts its energy and dies. So amount of energy is limited. And the last, that is the tail, which has fine thread like axial filament, arises from the posterior centriole and transverse the middle piece and the tail. So spermatozoa swim about by the vibrating their tail in a fluid medium in search of ova. Now the second gametogenesis, that is the oogenesis. Oogenesis takes place in the ovaries. Unlike sperm formation that starts at puberty, egg formation begins before birth but is completed only after fertilization. In the process of oogenesis, it consists of three phases. First one is multiplication phase, second is growth and differentiation phase, and the last is maturation phase. Now, in this picture, you can see the complete process of oogenesis. So, the first stage, that is the multiplication phase, where the certain cells of ovaries are larger than the other and have large nuclei. Then cells undergo mitotic divisions, producing undifferentiated feme cells, that is the oogonia or egg mother cell. Now, this oogonia, that is diploid structure under mitotic divisions to form primary oocyte, and it is the future ovum. Other oogonia except the primary oocyte forms follicular epithelium round the primary oocyte to protect and nourish it. That structure is young a primary ovarian follicle. Now, the primary oocyte remains at prophase of meiosis first until just before ovulation in an adult female. This is the first resting period in case of oogenesis. Now, the growth phase of the primary oocyte is very long, extending over many years. Its cytoplasm increases and the nucleus enlarges after puberty. In maturation phase, the primary oocyte after growth enters into the first meiotic divisions to produce two daughter cells. This is unequal divisions where one daughter cell is extremely small and where the other is large as the primary oocyte. Here, the small cell is called first polar body and the large cell is called the secondary oocyte. Here both are haploid structures. Then the secondary oocytes undergoing the second meiotic divisions which does not proceed beyond metaphase until a sperm enters in it. This is the second resting period. This is also unequal divisions to form large ooted or mature ovum and very small secondary polar body, and the first polar body may divide to form the two polar bodies. So, after the two meiotic divisions, one primary oocyte forms one haploid oocyte and the three haploid polar bodies are formed. This oocyte grows into a functional haploid ovum and its development takes 1 to 3 years. And after the fusion with male gamete, it forms the zygote that is diploid in condition. Here, the half number of chromosomes comes from the male and half from the female. So this is the complete process of the oogenesis. Now, in this picture, oogenesis involves three phases, that is the proliferative phase, growth phase and maturation phase, during which the primary germ cells progress to primary oocyte, secondary oocyte and then to mature oocyte. And the oogenesis differs from spermatogenesis in that it begins in the fetus prior to birth. Oogenesis is the differentiation of the ovum. So at first the primordial germ cells form primary follicle which forms secondary follicles. Here it shows many phases like the pre-antral follicular phase and antral follicular phase and ovulatory follicles, etc. And after that the ovulated eggs comes in fallopian tube, and after attachment of the sperm the fertilization occurs and the process starts of the embryogenesis. Now, the structure of ovum. This is a complete structure of ovum. The ovum is a rounded, non-motile and alacithal cell. It has abundant cytoplasm, that is ooplasm, a large nucleus, that is the germinal vesicle, a prominent nucleolus and surrounded by the plasma membrane, that is the oolemma. Here one thing, that is the alacithal cell or egg. What is the meaning of alacithal? Alacithal means absence of yolk. Now, the ovum has two poles. First one is animal pole and the second is vegetal pole. The ovum is enclosed by the two additional egg coat, that is inner zona pellucida and the outer corona radiata, which you can see in the picture. Zona pellucida is secreted by the ovum itself, thus called primary egg membrane. And the corona radiata is formed of radially elongated follicular cells called secondary egg membrane. So, the primary egg membrane is zona pellucida and the secondary egg membrane is corona radiata. It is very important from the examination point of view. Now, the differences, that means spermatogenesis versus oogenesis. So, in first, that is the multiplication phase, same in both and forms primary spermatocyte by spermatogenesis in male and the primary oocyte by the oogonium in female. So, the primary oocyte and spermatocytes are haploid in number. Second is growth phase, in which the primary spermatocyte forms secondary spermatocyte, which are same in size, and the primary oocytes form the secondary oocytes, which are also same in size. But here the condition is the one cell is larger in size and the one is small. The larger is known as known as oocyte and the smaller is known as first polar body. And then in the differentiation phase, the secondary spermatocyte forms 4 spermatids, but the secondary oocyte form one otid and 3 polar bodies, which are second polar bodies. Otid grows to form the ovum. So after a one spermatogonium, 4 sperms are formed, but from a one oogonium, one ovum is formed and 3 polar bodies are formed. So this is the differences between the spermatogenesis and oogenesis. Now, the differences in spermatogenesis and oogenesis. Spermatogenesis occurs in seminiferous tubules of testes, and the oogenesis occurs in ovaries. Spermatogenesis is the process by which sperms are formed from the sperm mother cell in the male, while the oogenesis is the process by which ovum is formed from the oogonia in females. In spermatogenesis, one spermatogonium forms 4 sperms or spermatozoa cells on maturation after the second meiotic divisions, while in oogenesis, one oogonium forms only one ovum and 2 polar bodies are obtained after the second meiotic divisions. In spermatogenesis, in male, spermatogenesis occurs throughout the life of human male, while the oogenesis stops at the age of menopause in human female. In spermatogenesis, polar bodies are not formed, while in oogenesis, 2 or 3 polar bodies are formed. Now, the similarities between the spermatogenesis and oogenesis. Both occur in the germ cells in the gonads. Both produce haploid reproductive units, that is the gametes, for the sexual reproduction. Both consist of 3 phases: multiplication, growth and maturation phase. Both involve changes in the nucleus as well as cytoplasm of the gametes mother cell. Now the significance of gametogenesis. It produces haploid gametes which by fertilization form a diploid zygote to maintain the chromosome number from generation to generation. It involves crossing over in meiosis first, which introduces variations in the gametes. And similarity of the gametogenesis in all animals indicates a basic relationship. Now, the differences between spermatozoa and ovum. So, there are some characters like, according to the definition, sperm is the male gamete and ovum is the female gamete. According to motility, sperms are motile while the ovum are non-motile. According to size, sperms are the smallest cell in the human body while the ovum are the largest cell in the human body. According to mitochondria, in sperm centrally located while in ovum is scattered in the cytoplasm. Cytoplasm in sperms present in very small amount while in ovum present in very large amount. Nucleoplasm is absent in sperm while it is present in ovum. Chromosomes contains X or Y chromosomes in sperms and contains only X chromosome in ovum. Centrioles are present in sperm while it is absent in ovum. Sperm is surrounded by plasma membrane while ovum is surrounded by egg envelope. In case of formation, one spermatogonium produces 4 sperms while one oogonium produces only one ovum. So this is the main differences between the spermatozoa and the ovum. Now, the differentiation of gametes. So at first, in case of the male, that means in spermatozoa. So first is the changes in nucleus. Here, nucleus becomes smaller but longer and the DNA condenses, RNA and nucleolus loss and the chromatin fibers become closely associated. In case of changes in Golgi apparatus, it forms the acrosome. In case of male, changes in centrioles. The centrioles shift behind the nucleus and come to lie one behind the other. In changes in mitochondria, it shifts behind the centriole and get arranged in a spiral around the axial filament. Changes in cytoplasm, the cytoplasm extends around the entire sperms or at the sheath. Changes in plasma membrane. It extends around the developing spermatozoa to cover it fully except the end piece. Next is differentiation of ovum. And in this case, first is changes in the nucleus. Nucleus enlarges due to increase in nuclear sap. And the second is changes in the cytoplasm. Here, the cytoplasm increases in amount and called ooplasm. Here, the ribosomes and mitochondria increases in number. Endoplasmic reticulums become more elaborate. So these are the differentiation at the time of gamete formation in spermatozoa and ovum. Now, the menstrual cycle. The first menstruation begins at the puberty and it is called menarche. The menstrual cycle in humans lasts 28 or 29 days. The days are numbered from the first day of the blood flow in the menstrual period. The cycle of the events from the first day of the menstruation to the start of the next menstruation is called menstrual cycle. And this ceases around 50 years of the age, which is known as menopause condition. So in the menstrual cycle, there are three phases. First one is follicular or proliferative phase, second is secretory phase or luteal phase, and the last is menstruation or bleeding phase. So here we will discuss every part in detail. So first one is the follicular phase or the proliferating phase. This phase lasts for about 14 days. Here, the anterior pituitary lobe secretes FSH, that is the follicle stimulating hormone, which stimulates the growth of the selected primary ovarian follicle and maturation of the primary oocyte in the follicle. Then, the FSH stimulates the follicle cells to secrete estrogen, which inhibits the secretion of FSH and stimulates the secretion of LH from the anterior pituitary. The LH induces the mature ovarian follicle to burst and eject its egg into the follicle tube called ovulation. The estrogen also stimulates the uterus to prepare for the implantation and nourishment of the fetus likely to arrive after ovulation. Under the influence of estrogen, the lining of the fallopian tubes is thickened and its cilia and their movements are increased. So these all the events occur in case of the follicular phase. Now the second, that is the luteal or secretory phase. This phase lasts for about 10 days. The peak of LH together with the prolactin hormone from the anterior pituitary lobe stimulates the follicular cells of the empty Graafian follicle to form a yellow body called corpus luteum. The corpus luteum secretes another ovarian hormone called progesterone and a small amount of estrogen. Now, the progesterone and estrogen continue the hypertrophy of the endometrial lining in the uterus and the fallopian tubes and maintain it throughout pregnancy. Progesterone is also necessary for the proper implantation of the fetus in the uterine wall. And the progesterone stimulates the endometrial glands to secrete a nutrient fluid for the fetus, hence the term secretory phase. Now, the progesterone inhibits the release of FSH so that it may not develop additional follicles and eggs. So, this is a complete luteal or secretory phase. Now, the third, that is the menstrual or bleeding phase. This phase lasts for about 4 days. If fertilization doesn't occur, the secondary oocyte undergoes autolysis and the progesterone inhibits the release of LH from the pituitary. Reduction of LH level causes regression of corpus luteum by the autolysis and a consequent fall in the progesterone level in the blood. The uterine lining dies due to deficiency of progesterone and is slugged off. Now the blood vessels rupture causing bleeding and the lining of fallopian tubes break down. The cast of uterine and the fallopian tube tissues, bloods that oozes from the ruptured endometrial blood vessels and the tissue fluid from the endometrial surface pass out through the vaginal opening called the menstrual or menstruation, and this is also known as weeping of uterus. It occurs after about 25 days and continues for 3 to 5 days. So this is the menstrual or bleeding phase. Here, In this picture, you can see the brief description of the menstrual cycle. In menstrual phase, which is for day 1 to 5, here the progesterone secretion is reduced. Endometrium breaks down, the cells lining the endometrium, secretions, bloods, and the unfertilized ovum constitute the menstrual flow. After this, the follicular phase, which is from day 6 to 13. And here, the follicle grows, FSH and estrogen secretions increases, endometrium starts to rebuilding. Now, next is ovulatory phase, which is on 14th day, where the LH and FSH secretion from the anterior pituitary attains a peak level and concentration of estrogen in the blood is also high and ovulation occur. And the last is luteal phase, which forms day 15 to 28. And here the corpus luteum develops and starts secreting progesterone. And here the endometrium thickened and uterine glands also develop. So this is the complete menstrual cycle for 28 days. Now, effect of fertilization. If fertilization occurs and fetus implanted in the endometrium, the trophoblast cells of the developing placenta secrete a hormone that is known as HCG, human chorionic gonadotropin. And this hormone, like LH, maintains the corpus luteum and the secretion of progesterone and estrogen by it. These two hormones check the breakdown of the endometrium of the uterus, and the absence of menstrual bleeding is the earliest sign of pregnancy. Now, the miscarriage. Premature degeneration of the corpus luteum is the common cause of miscarriage at about 10 to 12 weeks of pregnancy. And what is the pregnancy test? During pregnancy, HCG may be detected in the urine, and this forms the basis of pregnancy. After this, you can see here there are some differences in the menstrual and estrus cycle. So what is the differences in the menstrual cycle and the estrus cycle? Menstrual cycle occur in the primates only, while the estrus cycle occur in most species of mammals except primates. Menstrual cycle, there is bleeding at the end of this cycle, while in estrus cycle there is no bleeding in this cycle. In menstrual cycle, the broken down endometrium is passed out in the menstrual flow, while in the estrus cycle, the broken down endometrium is reabsorbed. In menstrual cycle, there are no cyclic changes in the sex erg of the female, while in the estrus cycle, there are cyclic changes in the sex erg of the female. In menstrual cycle, the female permits copulation at any time during the cycle, while in the estrus cycle, the female permits copulation only when in estrus or heat period. So this is the changes, this is the differences between the menstrual cycle and the estrus cycle. Now, the last topic, that is the artificial insemination. It is a technique to make a female pregnant by artificially introducing semen into the vagina. It is an application of our knowledge of the process of fertilization. It is commonly used in the farm animals. Semen of good quality male animal is collected, preserved by freezing and used when required. Now, what are the advantages of the artificial insemination? In this, the semen of good quality male animal can be used to inseminate a number of females. Preserved semen can be transported to distant places, excluding the need for sending the male animal there. And the transmission of sexual diseases are checked by artificial insemination. It is used in human beings for improving the chances of fertility. Here, a donor of a sperm can be husband or any other person. So, if husband is semen provider, it is called AIH, that is artificial insemination husband. And if the other donor is sperm provider, is called AID, that is artificial insemination donor. In this, if a man may be infertile due to insufficient number of sperms, weak or premature ejaculation, inability of penis to undergo erection and enter the vagina, or non-motile sperms, in all these cases, the semen of husband or donor may be collected, concentrated, and introduced artificially into the wife's vagina. It poses moral and legal problems, and the children produced by this technique are called test tube babies. So, this is the end of this video. Here you know about the spermatogenesis, oogenesis, structure of sperm and ovum, what is the menstrual cycle, and the artificial insemination technique. Now, the upcoming next video will be on the fertilization. So if you understand this video, please like and share it and subscribe to my channel Anupma Biology Classes. If you have any questions, any queries or any suggestions, you can ask in the comment section below. Thank you for the watching.